We have Dr. Nicholas Fernandez. After completing a postdoc in Avi Mayan's lab at Sinai, Dr. Fernandez joined the Human Immune Monitoring Center as a computational scientist. There, he works on developing computational pipelines, interactive web-based visualizations, and novel techniques for analyzing high-dimensional biological data. I'm delighted to present Dr. Nicholas Fernandez.
Hi, thank you for the introduction, and thank you for having me speak. And today I'll be discussing kind of a long title, high-dimensional single-cell data analysis using prior knowledge signatures, unbiased clustering, and WebGL data visualization. Okay, so here's a link to the slides if you want to see them, but I'll put the link up again later, but if you want to check it out, you're welcome to. So the talk will be broken down into three parts. First is just this discussion about data visualization from a broad level, because we've seen today, along with an enormous amount of information about the experimental background, there's also been a ton of different visualizations. So to just attempt to put everything into context, sort of an overview of data visualization and my opinions on where we should grow with it. Then I'll discuss building our analysis pipeline and toolset, sort of alongside a discussion of the case studies and future directions. Okay, so just to start, biological data is difficult to visualize. So part of the reason that that's the case is because it can be high dimensional. So you have bulk RNA-seq and proteomics where you have thousands of dimensions in bulk cells, flow cytometry where you have maybe like 50 dimensions but across millions of cells, and then single-cell RNA-seq and related methods like we've seen today where you have tens of thousands of dimensions but also tens of thousands of data points, tens of thousands of cells. So this can lead to like the familiar hairball we've seen, where here this is just some protein-protein interaction network, ball and stick network that is accurate, but it's not really interpretable at all. It's just a big blob. So we can sort of step back and just ask ourselves, like, can we visualize high-dimensional data as a broad question? And depending on who you ask, you will get different answers. And we can refine that question a little bit more, and we can say, can we do it without dimensionality reduction? Because we all know we can do it with dimensionality reduction. Furthermore, can we work with hundreds to thousands of dimensions, or can we work with hundreds to thousands of data points simultaneously? Does it even make sense to do so? Some people ask, you can do it, but it doesn't make sense. Like kind of like this figure, yes, you can look at an interaction of 10,000 proteins and point out all their interactions, but what are you getting from it? Like, will the result be useful, right? So it's not just a question of can it be done, but what's the point? Why do it anyway? So this topic was discussed in relation to flow cytometry data and mass cytometry also. And in a recent paper from Nature Immunology in 2016, where they were discussing how the field is being blessed with the so-called curse of dimensionality. And the curse of dimensionality refers to many things, but one of the things that it refers to is just the exponential increase in state space of a system when you add more dimensions to it. So this figure to the right here, taken from the paper, shows in a simplified state, let's say you're dealing with surface markers on a cell. If you're looking at one marker and you have just a simplified high or low state, you just have two states in a one-dimensional system. You go up to two states, and now you have a square, you have four states. When you get up to three separate markers, you're now in like three-dimensional space, even though it's just binary. Now you have 8 markers. Once you get up to 40 markers, the number of possible states is 1 trillion, over 1 trillion. So it's hard to think about that state space, and it's hard to work with. Even though it doesn't sound like it's that high dimensional, even in a simplified binary dimensional space, it's actually really enormous. And they're using a piece of modern art as a surrogate representation of a hypothetical, unvisualizable 40-dimensional data set. So we definitely know where they stand. They think, the case they're making here is that you can't directly visualize this high-dimensional data. And one of the things that is very commonly done, we saw in a lot of really good talks earlier, is dimensionality reduction. And the basic idea is to project high-dimensional data into fewer dimensions, usually 2 or 3, that can be easily visualized. And 2 very common techniques are principal component analysis and T-stochastic neighbor embedding. And the techniques work very well, and this is an example here taken from a really great blog post about visualizing the MNIST dataset. And here you're looking at a TS&E projection of handwritten digit images. So each image can be thought of as a high-dimensional data point, where each pixel in that image is actually a dimension, and the pixel level is the value of that dimension. So a handwritten image of this sort of low-resolution image is actually exists in a 700 dimensional space, more or less. So if you do, if you want to visualize that as like scatter plots, with perpendicular dimensions, it can't be done. You stop at 3. You can't get any higher. So TISNE, in this case, does a really excellent job of reducing those high dimensional points onto 2 dimensions, and it arranges them in such a way that they're close to their local neighbors. So it does a good job of finding like nonlinear neighborhoods. And this is done in an unbiased fashion, but the colors are overlaid for each digit identity. So this is just a toy example, but it does a pretty good job, or you would say very good job, of like finding groups of numbers that are actually the same number. So finding them together. Problem, however, is that it lacks interpretability. We don't know anything about the pixels. We don't know from this figure alone. We don't know why 2 pixels are actually, or 2 images are actually similar to each other from this image alone. We have no idea. So that may not be a big deal if you're dealing with image data, but if you're dealing with gene data, you probably care a lot about your actual dimensions. Okay, so we could ask ourselves, or we could say to ourselves, well, we're told that this image is a high dimensional data point, right? We can clearly see that. It's an image. It's a handwritten image. So maybe there's some hope that we can actually visualize high dimensional data. So if we take a step back to data at its, you could argue it's more fundamental state. When you usually get it from somebody, it's in a spreadsheet, right? So you could make the argument, it's kind of a trivial, silly argument, kind of, but a spreadsheet just allows you to directly visualize high dimensional data. It's not pretty, but what it lacks in like beauty, it makes up for in precision, because you know what each number is. And the way you can think of it as a spreadsheet is each column is a data point, and each row is a dimension. So here we're looking at somebody's budget over January, February, March, 3 data points, 3 dimensions for phone, insurance, rent. But obviously you could add as many dimensions and as many data points to a spreadsheet to have, and you're able to like directly visualize that data. The pros are that it's very interpretable. You can go up to hundreds or thousands. Problem is it's slow to visualize, and it's barely qualifies as a visualization. But if you just replace the numbers with colors, then like bam, you get a heat map, right? You go from this Excel sheet to something really beautiful here, and you have a real visualization. So clearly, it's not unvisualizable data that we're able to visualize it, right? So mind blown. And that was a funny gift. So okay. So obviously a lot of this is like very clear to you guys, but just want to contextualize everything together. Okay, so now we move on to real heat maps, right? So a heat map, which is basically just a glorified spreadsheet. In a heat map, the thing that's commonly done is to arrange your data points in a way where they are similar, placed to data points that are similar to one another, and the same with dimensions. So in this case, it happens to be that we have samples here as rows. It's just a toy data set. And then you have expression of genes as columns. So you're actually, an advantage of a heat map is that you're giving equal weight to your data points and your dimensions. So you're not preferring putting one on a pedestal and the other like gets lowered. Like it's equal weight for both of them. And so by positioning similar data points and similar dimensions next to each other, you start to see structure in your data. And then you can show the distance between these data points using a dendrogram, which is this nice visualization that shows you like this cluster and this cluster are separate, and the level at which this dendrogram branches out tells you how different they are on some distance metric. Could be Euclidean distance, could be anything. The problem with static heat maps the good thing is that for small data sets, they work just fine. But once you get to larger and larger data sets, everything becomes smaller and smaller, and it kind of gets us back to the point where we're almost as bad as that hairball. I mean, we're better because it's interpretable and you can see some structure in it, but you don't know what anything is, right? So it's not very useful. So that kind of brings us back to our initial state. So we have something that is really close, but it needs something else to get it to be more useful for the size of data that we're dealing with. So for that purpose, this sort of started me in my journey of like web development and data visualization while I was a postdoc in Unbiased lab here at Mount Sinai. And we started on a project that we were calling Clustergrammer. So our goal was, broadly speaking, to extend the capabilities of heat maps, enable complex interactive data exploration, make it user-friendly and shareable, and also add some biology-specific features, but without making it tied down to some particular data set type. And the vehicle we chose was the web browser. So to utilize the browser so that it would be easy to share with people and put online, that kind of thing. So Clustergrammer was built with a library called D3JS. It's data-driven documents. It's a very popular data visualization library authored by Mike Bostock. And it was actually, you've probably seen visualizations from D3 if you've looked at the New York Times over the past 5 to 7 years, because they utilize it a lot. And so it's a really good library for building customized and interactive visualizations, and everything's at a low level. Everything's you build it up from tiny little shapes, which is what we need for our custom visualization. Now the back end, the clustering data analysis, we chose to use Python because Python has an extensive numerical and scientific ecosystem pretty much, and it's also a general purpose language, so you can build web apps and Jupyter notebooks and that kind of thing. And we also built a small web app that accompanies the cluster network project, which I don't explicitly mention here. So I'll just quickly, I'll just quickly show an example of the cluster grammar heat map. So this is just a toy data set with some lung cancer cell lines taken from the Cancer Cell Line Encyclopedia, just a small snapshot of the data set. That real data set is much larger. So you have a subset of kinases here as rows, and if you hover over them, it'll tell you the full name and the description. It's actually talking to an API when you do that. So if you load your data, it'll help you, because nobody knows all the gene names on off your hand, probably not. And so you can zoom in, you can reorder, so we see structure here, and we can we have a sort of compactified dendrogram, so we can choose larger and smaller clusters. So we can find here a cluster of cell lines, and we can see the breakdown of categories in that cluster. And we can also, it's also performing the Fisher exact test to tell us the likelihood that you would have picked that many data points from a particular category from that cluster. So it immediately allows you to compare data-driven clustering to prior knowledge in the form of categories. You can also reorder, like I was mentioning earlier. So if you rank everything by sum, everything is animated, and you can see other structure in your data. You can all of a sudden see that this cell line is higher for kinases up here than these, and that sort of thing. And then you can reorder by categories. There's a lot of flexibility, and importantly, it lives on the web, and it allows you to, just generally speaking, visualize high-dimensional data, share it with people. So that's JavaScript library. The next thing is that we wanted to do was bring it a lot closer to the data analysis end of it, so the Python end. So in the early days of the project, I used to make a custom web page for somebody and send it to them, and everything was separate. The Python was separate. But I heard that Jupyter Notebooks was developing systems to help you integrate JavaScript with Python. And so background, for those of you that are not familiar with a sort of big project called Jupyter, it's one of the main things with the Jupyter project is a Jupyter notebook, and that's at the core of this project. So the idea of the project is to allow people to perform data science in a graphical user interface, but also to allow communication between computational people and experimental people, and to allow you to combine images, code, and just free text all in one document, so everything's combined together, and also to facilitate reproducible research and that kind of thing. So a Jupyter Notebook is, there's a lot of different forms of it, but you can kind of think of it as a hybrid web app, because you can run it locally. You can run a local Jupyter web server and do your data analysis there. And you can also share that on the web, and it can also even be run on the web, which I'll show you guys also. So to take the JavaScript library we got and integrate it into Jupyter, we used a cookie cutter template. And this project was actually I recently presented it at JupyterCon 2018, just last August. And I think I should have an example. Let's see. Yeah. Okay. So one thing you can do is one really powerful feature of the library is that you can share your notebooks on the web on a. Okay. All right. Okay, so this is a notebook that's hosted on GitHub that's being viewed through NVViewer. But basically, for your purpose, you can imagine that we perform some data analysis, we pushed it up to GitHub, and we want to share it with the world. So you basically get a free web page with an interactive visualization on it. And this is just on the web. So you have it's not an executable notebook, but you can interact with the interactive components of it. And you can see how it was actually run and try and understand what happened. And then you could also go to the GitHub repo and get the code for the notebook and download the whole thing and reproduce the research on your own. So So this is just a tweet, just somebody that's kind of popular in the data analysis field saying how huge it is that you can now embed Jupyter widgets in a notebook online. So that's really cool because you can basically very easily, in maybe 10 minutes, create a web page on the web, like real web, with interactive visualization. The next thing is actually executing those notebooks. Because if you want to rerun the analysis, like if you don't trust what I did or you want to change something, you have to download all the libraries, make sure everything's installed properly, make sure you have the same versions of everything, and it can be time consuming. So there's a service called Binder, actually is like an amalgamation of a bunch of smaller services, but for our purposes, it basically allows you to take a public notebook and rerun that analysis on the web, in the cloud, so it allows extremely easy, reproducible research. And actually the team that identified the gravitational waves recently from LIGO, their notebooks are on GitHub and they can be run on Binder, so you can actually rerun their analysis on the web for free, completely reproduce their results. So the latest thing we're working on, the WebGL part of the title, refers to the rebuild of cluster grammar on using WebGL, so we can handle much bigger data sets. So WebGL is a language that runs on the web, and it utilizes the graphical processing unit. And basically, it allows the GPU has many processors that are not that fast, but it can run things in parallel. So for certain tasks like image processing, it's much faster than a CPU. And for our purposes, it enables visualization of large data sets. And there's a lot of libraries out there, but I'm using a library called Regal. So just to show you what it looks like, so WebGL is usually done for like 3D stuff, right? And they have nice examples that you can actually go and play around with. So I can do that. Yeah, so you can like change your code around. And the thing is, all this code was needed to just draw one like red triangle, right? So it doesn't sound like a good library, right? But then all this code was needed to draw all these triangles. So I'm going to attempt to do something fun real quick. I'm loading up this example. It's a 3D, yeah, it's kind of cool. So it's this, this is like the default example for 3D visualization stuff. So what's cool is it's calculating the reflection of the image in real time on this bunny, and it's making it look like 3D. But I'm just going to try and quickly do something that alter it. So you can alter this code in real time, and it will rerun, like if you're lucky. And so I'm just bringing in this time-dependent parameter, and I'm hitting the position of the bunny in real time. So now the bunny should be like shrinking in size and growing in size once it's done rendering. Let's see. You saw it for a second, right? See if that works. I know. Okay, there we go. So now it's just growing. And if you look close, you can see the thumb of the person holding the camera. So what's fun, you can learn Regal and actually play with this stuff on the web in the real time. And this new laptop has like a fairly good GPU, so it can handle this kind of thing. So that's what a lot of WebGL is done for, or GPUs are done for graphics. Also lately, cryptocurrency, that kind of thing. Anyway, so now, and just coincidentally, there was a recent article published about using GPUs in scientific data analysis, just like 2 days ago in Nature commentary. And basically, it summarizes how GPUs are done for visualization using libraries like WebGL, but you can also use languages like CUDA and also libraries like GPU.js, which allows you to utilize the GPU for general purpose data analysis in JavaScript. And won't go into that, but so we're obviously using WebGL for data visualization, but one thing we're looking at for the future is to utilize WebGL for just dealing with big matrices and doing data analysis. So with all that sort of intro, we'll now move on to the 4 case studies that will hopefully show you the ideas behind our data analysis pipeline and also show you some case studies. So the first case study is a public, I'll just go over them real quick. So the 4 case studies. The first one is a public data set from 10x. It's 2,700 PBMC cells, single cell RNA-seq. The next case study is an internal site-seq PBMC single cell protein and gene expression data set. So we're all very familiar with site-seq now. The next data set is a hashtag multi-tissue scTECH data set to just demonstrate how we can combine multiple tissues together. And the last data set is a data set where we identify T cell clonotypes from VDJ sequencing, and we also have the gene expression data to show a quick example of what we can do with that. So here's a link if you want to follow. You could write it down, or you can check it out later. But this demonstrates our first case study, and it also demonstrates what I was mentioning earlier, Binder. So the exciting part about this is that if you click this link, it will take you to Binder, and it will actually, everything works properly, it will run the notebook and all the analysis from actually just GitHub. So the code is public, it's hosted on GitHub, the data is public, hosted on GitHub, and now it started up a Jupyter notebook. So this is a real Jupyter notebook that we can do like actual data analysis on, right? And we're running Python 3, you can see up here. And it takes a second to run, so I'll start it and just explain as it's running. So I'm telling it to run all the cells in the notebook, and it's just going to do a quick kind of toy analysis of this common data set and show sort of our approach to things. So here, we're first loading the libraries we need, we're loading the data, and you can see you've got 32,000 genes and 2,700 cells. And then we're just removing ribosomal mitochondrial genes here. Code is kind of messy, and then we're keeping the top 5,000 expressing genes here, just to drop genes with low expression, and then we're taking a subset of the data where we're then keeping the top 1,000 genes based on their dispersion, so the variance divided by the mean, and then just taking a random subset of those cells, taking 1,000 cells. So just sort of simple like operations, and we just want to see what we get, see what the data looks like using if we just cluster that data in an unbiased manner. So should be loading up. Yeah, okay. So now we're using clustergrammer, and we're using the new WebGL clustergrammer. So we have this is what the new widget looks like, and you might need to like resize your screen if you try this because it's kind of large. But so now we're looking at a matrix of 1,000 randomly selected cells and the 1,000 genes with the highest dispersion, and we're just doing unbiased clustering on those. So 1,000 by 1,000, that's like a 1 million matrix element that is being calculated on the cloud. So all this is running on the cloud with like limited resources. So just the fact that it works initially is already impressive to me. But now you're able to easily interact with this large data set because we're using WebGL. We can easily like peruse the data. So if we zoom into this red cluster over here, we see that these cells here with their barcodes are high expressing of granzyme, perforin. So we can make some hypothesis of what the cell type is. We zoom over here, we can see that these are high for like HLAs, right? But this isn't that useful because we could go and try and label the cells, but it's kind of difficult to do that manually, and we have like 1,000 of them, so it'll take too long. And you can identify different clusters here, but unlabeled data is not that useful. So one way we can quickly label the data is by bringing in some prior knowledge. So we just brought in the gene expression signatures from CyberSort, which is a public data set that was published previously. So these are their signatures, gene expression signatures of different cell types. So they have these broad cell types. So we're just taking these signatures and we're using that to label our cells. And this all already ran in real time. So we took that same 1,000 cells and we labeled them. So now CyberSort is telling us that these cells that are highly expressing granzyme, perforin, so forth, are NK cells, right? Which makes sense from what we understand, and it agrees, like makes logical sense. And then we're also seeing that it's labeling these as B cells, and then we can go in and like compare now our data-driven clusters, which is what we see here. So if we zoom into this, we see that this cluster here, is composed of 114 NK cells. 77% of that cluster is NK cells, and the odds of that happening by chance are like astronomically low. And so we can now see that even using like a very like kind of brute force cookie cutter attempt, we're already seeing that the prior knowledge agrees with our data-driven clustering for these cells, and we're able to do this. And this analysis can be reproduced by anybody by clicking that link and rerunning it. So it's getting to the point where now you get a free web app on the web using these services. Okay, so that's that first example. The next example is a scTECH. I won't go into it because it's been discussed many times, but the takeaway is that we're able to measure 30,000 genes, and at least in theory, and then about 50 proteins, and we're able to do that for thousands of single cells and from the same cell. So from one cell, you get protein level and gene expression level. So from my perspective, I'm given two matrices now, right? But they're from the same cell. So now we have this specific data set that is unpublished, is from a PBMC, human PBMC. And it's actually a lot of cells because it was multiple samples hashed together, but we have over 15,000 cells. And I'm just going to look at both components of the data set. So I'll start with the protein level markers here. Okay. So okay. So I'll try and go real quick. So so this is an example of looking at 5,000 cells in what we're calling ADT space. And we can see here how the cells cluster. So we can like zoom in and out of these 5,000 cells. You got 37 markers here. And this is unbiased clustering. So we can see that the unbiased clustering, and then up here the categories, these were manually gated by Adib. So we can see there's very good agreement between the manually gated categories and our unbiased clustering. And then we could do the same enrichment analysis trick where we see this, the agreement between our prior knowledge and our data here. We can also like reorder based on the gating there. And WebGL is able to handle a lot of data, so it shows us, in this case, we have the doublets listed here as black, and we can see that they're kind of like this big red blob here. We'll move on to the gene expression data. So now if we take those same labels from ADT space, overlay them on gene expression, and just unbiasedly cluster 5,000 randomly chosen cells based on the, I think it's top 500 genes based on variants, we see that, again, there's very good agreement between the surface marker data and the gene expression data. And then we can zoom in and see like the specific genes that are being differentially expressed in the cell types. So this figure here is basically showing the gene expression data and the ADT data agree pretty well on a broad sense, and using very simple metrics. Yeah, and then one other small thing, if we can like zoom into CD8 clusters, and we can look at subtypes of CD8s here. So here we've got naive CD8s over here, and then CM CD8s over here, and we can see that we're getting clustering of CD8s by their subtype also. So it gets us down even to lower level granularity, and then we can like reorder, and then it just sort of see what's happening. And then down here, we can also do the reverse. We can go from categories to signatures. So before we got a signature and we made categories from it, now we can just point to a category and say make a signature, which is what we did here. And now we have these cell types. So these were categories prior knowledge from the ADT space. And we're now getting the differentially expressed genes for these different cell types. So we can zoom in and we can start to, we could, for instance, do enrichment analysis on that data set if we wanted to. Okay, let's get through that. So that was that. And then this is a quick example for a scTECH data set where 5 tissues were hashtagged together. What we have here, yeah. Okay, so up here we have 2 categories for each cell. We know which tissue it came from, and we know which cell type. So you can see that you get clustering based on both, but it appears that the dominant clustering, at least visually, is from the cell type, like which sort of biologically makes sense. But let's see, we'll try and order by tissue, but you can still see that if you reorder by tissue, you still have this kind of like block by tissue. Despite the fact that you're mixing all the cell types together, you're getting these blocks. And then you can also see that some tissues are really have high level of a particular cell type as compared to other tissues. So this is just a very unbiased way of looking at our data. You order by one and then the other?
Yeah. By tissue, then cell types and the tissue.
Not on this figure, but the best way to do it would probably be to take a cell type and then reorder that by tissue, like break it apart. And then here we just constructed signatures from those cell types. I won't go into that, but to just show you the sort of symmetry of how we're trying to do things. And then the last example is just a T cell clonotype and gene expression. This is pretty quick also. So here we're looking at, sorry, let's see how many. It's also about 2,000-ish T cells. We dropped all the T cells that they didn't have like a valid VDJ sequence. And we're showing just based on I believe it's top 500 genes variants. So we're labeling these T cells and we're labeling with their clonotype. So you see there's not too much clustering based on clonotype, at least in this view. and you're not really seeing too much significant clustering happening here. So what I try to do, and this sort of underscores, and show you how many different clonotypes there are, but it underscores the importance of like picking the right dimensionality and also like filtering your data. So if we just drop all the clonotypes except the top 10 most abundant clonotypes, and then we recluster the data just based on the top 250 variable genes, all of a sudden we start to see clustering by clonotype. So you can see here that like this cluster here is going to be enriched for this yellow clonotype. So we're starting to see the differentially expressed genes based on that, and then we can construct a signature from those clonotypes. And then here, this is going back to the old widget, the SVG version, but this is just showing a feature that's kind of fun, where this is the same data. This is the signature of these clonotypes. So if we want to be able to bring in some kind of if we want to be able to bring in prior knowledge, we can do that using our web API. So we can talk to an enrichment analysis tool called Enricher, and we can compare these genes to prior list genes, and they can show us which genes are related to which phenotype. And in this case, it's kind of a little bit uninterpretable, but yeah. So based on time, I'll move on, and I think that's about it. Yeah, the project is public. Everything's open source. You can check out all the documentation. And I would just like to acknowledge everyone at the Human Immune Monitoring Center, especially Miriam Murad, Adib, Singi, Sasha. Melanie did all the pre-processing for all the single-cell data, and he does an extremely great job. And everybody at the HIMC, also Avi Mayan in the Mayan Lab, everybody at the Mayan Lab, especially Greg Gunderson, Michael McDermott, Matthew Jones, and my colleague and wife, Dawn Fernandez, in the John O'Reilly Lab, who we're also collaborating with. And thank you for the opportunity.